All right, let's jump right in. Welcome to this explainer, where we're going to unpack what I like to call one of nature's most high-stakes biological heists. Every single day, right beneath our feet, plants are desperately trying to unlock a vital, life-sustaining treasure from the microscopic vaults of the soil. That treasure is iron, and as we're going to see, plants have actually evolved two radically different, highly sophisticated strategies to pull off this heist. We're going to break down exactly how they do it, step by step.
But before we look at the suspects, we got to talk about a massive contradiction. If iron is literally one of the most abundant elements in the Earth's crust, why are plants constantly starving for it? It's a genuine paradox. Iron is absolutely non-negotiable for a plant's survival. They need it to synthesize chlorophyll, the green pigment that makes life possible. It acts as this crucial cofactor for enzymes. It's a core piece of the electron transport chain in photosynthesis, and it's even required for DNA replication. The plant needs it desperately. The iron is physically sitting right there in the dirt, but the plant simply cannot access it. It's totally locked away.
So why is the vault locked? Well, it all comes down to the chemistry of how iron exists in the soil. It mainly shows up in two different valence states, and let me tell you, they are not created equal. First, we have ferrous iron, or Fe2+. This is the good stuff. It's highly soluble, plants can easily absorb it, and it thrives in acidic or flooded soil conditions. But then we have ferric iron, Fe3+. This is the stubborn, locked-away form. It has terribly low solubility and mostly exists as solid, insoluble rust, ferric oxides, especially when the soil pH creeps up over 6.5. So if a plant is growing in neutral or alkaline soil, it is literally surrounded by iron it just cannot use.
Now, what happens if a plant botches this heist and can't get to that iron? Well, it's pretty devastating. If a plant can't access that stubborn Fe3+, it suffers from a condition called chlorosis. Because chlorophyll synthesis gets blocked, the young leaves start turning pale and yellow while the veins might eerily stay green. Inside the plant cells, the chloroplast ultrastructure is literally disrupted and the whole photosynthetic machinery just shuts down. No photosynthesis means ATP synthesis drops, growth grinds to a halt, and crop yields absolutely crash. For the plant, accessing iron is quite literally a matter of life or death.
Let's meet our first group of biological safecrackers. High Screw 1. Strategy I. The reduction method. This reduction method is the main playbook for non-gramineous plants, so your dicots, like Arabidopsis and soybeans. They use a very brute force approach to bust into the iron vault, breaking it down into three distinct steps. First up is acidification. The plant actively pumps acid out into the soil around its roots to essentially melt the vault doors. Second is reduction. The plant uses a chemical reaction right on the root surface to forcefully strip an electron away, converting that stubborn Fe3+ into the highly usable Fe2+. And finally, step 3, transport. Once the iron is in that usable ferrous state, the plant quickly shuttles it right across the cell membrane and safely inside the root.
Every good heist needs specialized tools, right? First up, we've got the H+ ATPase, specifically one called AH2. Think of this as the acidic drill. It's a localized pump on the plasma membrane that constantly blasts protons into the soil, lowering the pH and solubilizing that rust. Next is FRO2, the ferric chelate reductase. This is our lockpick. It does the heavy lifting of reducing the iron to Fe2+. And actually, this is considered the limiting, absolutely most critical step of strategy I. Finally, we have IRT1, the iron-regulated transporter. This is the high-affinity getaway car. As soon as FRO2 creates the Fe2+, IRT1 grabs it and mediates its transport straight into the root cells. And what's wild is that AH2, FRO2, and IRT1 can actually physically link up to form a supercomplex, making this whole operation incredibly fast and efficient.
But what if the soil pH is extremely high and proton pumping just isn't cutting it? Well, Crew 1 has a pretty clever backup plan. They deploy the coumarin assist. The plant roots will literally secrete these phenolic compounds called coumarins out into the soil. These coumarins act like chemical sponges, mobilizing that highly insoluble Fe3+ and converting it into a soluble chelate. Once it's been mobilized by the coumarin, it can be dragged back to the root surface where our lockpick, FRO2, can finally get to work on it.
All right, time to meet Heist Crew 2, Strategy 2, the chelation method.
This chelation method is the go-to strategy for gramineous plants. We're talking grasses, which includes massive global crops like rice and maize. If strategy I was a brute force safecracking, strategy 2 is a stealthy, high-tech biochemical net. Here's their slick three-step operation. Step 1.
Synthesis and secretion. The roots manufacture specialized chemical claws and secrete them out into the dirt. Step 2. Chelation. These chemical claws have an incredibly strong affinity for iron, and they bind tightly to the Fe3+, wrapping around it and literally pulling it out of the soil particles. Step 3. Transport. The plant doesn't bother absorbing bare iron, it just reels in the entire claw, with the iron trapped safely inside, straight across the root membrane.
So, what exactly are these magical chemical claws? They're called phytosiderophores, or PSs for short. These are low-molecular weight non-protein amino acids, molecules like mugenic acid. Think of them as the perfect stealthy claw machine. They have these 6 functional groups that perfectly align to grab Fe3+, forming an incredibly stable octahedral complex. By doing this, the grasses completely bypass the need for an acidic drill or a chemical lockpick. They just cast their nets and capture the iron whole.
And just like the first crew, Crew 2 relies on some incredibly specialized transporter proteins to pull this off. First, you have Tom1. This is the efflux transporter. Its entire job is to push those synthesized phytosiderophores out of the root and into the hostile soil environment. Once those PS molecules have successfully captured their Fe3+ targets, they got to get back inside. That's where yellow stripe 1, or YS1 and YSL transporters come into play. These are specialized membrane doors that only open for the complete, stable Fe3 phytosiderophore complex, pulling that captured treasure securely into the root.
But wait, here's the twist: blurring the strategic lines.
If you thought biology was going to keep these two strategies neatly separated in their own little boxes, think again. Nature is fiercely resourceful, and when survival is on the line, plants absolutely will steal from each other's playbooks. Our strategy I non-graminaceous plants, the dicots, can actually be opportunistic scavengers. If they're growing next to grasses, they can essentially steal the grass's hard work, directly absorbing the Fe3+ phytosiderophore complexes that the neighboring grasses secreted. No way, right? On the flip side, take grasses like rice. Rice often grows in flooded, low-oxygen conditions, where that highly soluble Fe2+ is naturally abundant. So instead of wasting energy building those complex phytosiderophore claws, rice can temporarily switch playbooks and use the strategy IRT1 transporter to just absorb the Fe2+ directly. And as a massive compensatory backup plan, when their main transporters fail, both types of plants rely on N-ramp transporters to facilitate emergency, low-affinity iron uptake. It's a total free-for-all down there.
So we can see that plants are already adapting, mixing, and matching these incredible cellular mechanisms just to survive. Which brings us to a really provocative question, and something to chew on as we wrap up. Could we actually bioengineer our vital crops to permanently use both strategy I and strategy 2? Imagine a supercrop that possesses the acidic drill of Arabidopsis combined with the stealthy chemical claws of rice. If we could successfully merge these two biological heists into a single organism, we could potentially grow robust, iron-rich crops in the most hostile alkaline soils on the planet. We could eliminate plant iron deficiency and the massive agricultural losses it causes forever. It's a fascinating puzzle for the future of agriculture, and it all starts with understanding these microscopic operations happening right beneath our feet.